Assalamualaikum warahmatullahi wabarakatuh and very good morning. Today we will proceed our lesson, science lesson, by discussing about the germination of seeds and the factor that affect the germination of seeds. Last week we have discussed about the structure of seeds and its function. So based on that, we now will proceed about and we will discuss about how seed is germinate. So now, from the discussion that we have made before, that a seed that contain the most important part is cotyledon, radicle, and plumule will play an important part in the process of germination. So during the germination of seeds, and what you have to know that from seeds, so plant is produced. Yeah? So you have to know that from a plant itself will be produced from the growth of the seed or germination of seed. So you have to know that. So very important for you that I repeat again that plant is coming from the growth of seeds. So we will look into what is exactly happened during this process of germination of seeds. So now, during the process of germination, the testa, we can look into this text, will burst, and then the radicle start to appear, and grow downwards into the soil to form roots. While the plumule will grow upwards to form a new shoot. So this is what we have discussed, and I have explained to you last week about that radicle will grow and form a root, while plumule will grow and become a new shoot. So during this process of germination of seed, cotyledon will have two conditions. So we can divide the germination of seed into two types. The first one we call it as a apigeal germination, and the second one we call it as a hypogeal germination. So what is the difference between apigeal germination and hypogeal germination? Okay, apigeal germination is germination of seed in which cotyledons are carried out of the soil. Okay, so this is the important definition for apigeal germination. So, in certain germination, for example, in this apigeal germination, cotyledons will be carried out of the soil. So, as shown here in the diagram, so this diagram shows about the apigeal germination. So the first part here, when we plant a seed into the soil, and then radicle will start to grow to the downwards, and then it will grow furthermore to form a root, and then the cotyledon will carry it out of the surface of the soil, and then it will become like this, and the position is at the upper parts of the surface of soil. So now from here, plumule will start to grow and will become shoot to form a leaf and stem. So now we can look here that cotyledon that carry out from the soil start to decrease its size, because as we know that cotyledon contains food that supply for the growth of this seed. So we can see this is what we call as a epigeel germination. So the diagram below shows about the hypogeel germination, in which during the process of seed germination, the cotyledon maintain its position in the soil, and then, as we have discussed, the radicle will grow to the downwards to become a root, and the plumule will form a stem and leaf like this one. So this is the how the process of seed germination. So it can be divided into epigeel germination and hypogeel germination. So that is the important thing in this explanation about how seed germinate to produce a plant. So after this, we will look into a video, several videos about several type of seed, how they growth, how they germinate, start from the early stage until to form a new plant. So after this, we will look into this video. Okay, so this is the first video about how the seed germinate. So now this is seed. So we start with the production of radicle that grows to become root. So you can see based on the day taken here. Okay, exactly this experiment, they record the process of seed germination from day 1 until it form a new plant. So we are very fortunate, very lucky to have this video, and we can see the process of seed germination by watching to this video that had been recorded by scientists. So from here we can know, even though it takes a long time, more than 20-30 days, so we can see very beautifully how seed germinate. So now we look into this plumule grows to become stem and leaf. So you can see the process of seed germination. Now day 35. So you can see it takes a long time for every seed to produce or to become a new plant. Day 40. Okay, now we look into other type of seed. This is a watermelon seed. Soil into the soil, and then now you can see from here, this is a radicle that grows to become root, and this one plumule now step by step will produce to become a leaf and stem. Day 14, 15. So this recording activity required a lot of passion to take this production or growth of seed. Day 25. Now this is a Chapoica. Will become a new plant through this seed germination. So you can see water level decreases. Root is formed. Sorry, this is a potato, sweet potato. Okay, right now this one, this is sunflower seed. Okay, okay, now we look into tomato. Okay, tomato fruit cut into pieces, and then inside the tomato we have seed here. Okay, you can see seed here. So we want to plant the seed, and you can see from here it will produce a new plant from a seed, from the tomato seed. Okay, pour water. Day 2, 3. Okay, you can relate to the day. Day 5, it start to grow. We cannot see inside the soil to watch how the radical grow. By the way, radical will grow first, and after that the plumo to form a leaf and stem. So you can see God Almighty have created this very beautiful creation. Okay, after we have watched how seed germinate, now we will look into what is the condition required for the germination of seeds. So from this text, we want to know what exactly how seed can germinate. What is the condition required by the seed to germinate? So in conclusion, we can say that seed require three factors given here. The first one is water. So seed require water. The second one is air. And the third one is temperature, suitable temperature. Okay. So these three factors, these three conditions must exist simultaneously. So if any of these condition is absent, so seed will not germinate. So that is the important concept that you must have. Means that in order for seed to germinate, so these three conditions must exist simultaneously, yeah, to make sure that seed to germinate. So very easy, just remember water, air, and suitable temperature. And the suitable temperature for seed to germinate normally in room temperature. Okay, so this is a suitable temperature because most of the seed cannot germinate in a cold, cannot germinate in cold temperature and also in hot temperature. So the suitable temperature for seed to germinate must be in room temperature. Okay, so that is the condition required for the germination of seed. So now, in order to prove these three conditions required for the seed to germinate, so we have experiment to conduct. So experiment 4.1, experiment 4.1. So in this experiment, so based on the format in conducting an experiment, so we must have problem statement, we must have hypothesis, we must have aim or objective of this experiment, we must have variables, we must have materials and apparatus, procedure, and the observation or the result of this experiment. So we will discuss about this experiment right now. So please pay attention. So this experiment, the aim is to determine what is the required condition for seed to germinate. So we want to know what is the required condition for the germination of seed. So the problem statement here, okay, are air, water, suitable temperature necessary for seed to germinate. That is problem statement. And hypothesis for this experiment is seed needs air, water, and suitable temperature to germinate. Okay, so the aim I have explained to you just now. Okay, what is the variable for this experiment? Manipulated variable is germination condition. Germination condition, that is the manipulated variable, whether water, air, or suitable temperature. Responding variable is seed germination, and constant variable is type of seed. Okay, now materials and apparatus use boiling tube, cotton wool, water, cooking oil, sugar, pepper, green beans, and refrigerator. Okay, so what is the procedure? The first one is we have to prepare. Okay, we have to prepare four boiling tube. Label A, B, C, and D. So we have to prepare four boiling tubes as shown below. Okay, so what is the condition for every boiling tubes? Will have their own condition to show about the required condition of seed germination. So the first one, boiling tube A. Boiling tube A. Okay, what is the condition for this boiling tube? Okay, all the boiling tube will be covered with sugar pepper. Okay, this sugar pepper is exactly black in color. Okay, and then we put wet cotton wool. Wet cotton wool here. and then we put seed on top of that. Okay, so this is for boiling tube A. For boiling tube B, we put dry cotton wool, and then seed on the surface of this cotton wool. So this is the B boiling tube. So next, boiling tube C. Okay, boiling tube C, we have cold boil water. So you must focus on this one, boil water that have been cooled. We put seed inside the boiled water, and then on the surface of this boiled water we put cooking oil. Yeah? So here cooking oil and sugar pepper. Every tube will have this, will be covered with sugar pepper, that black in color. Okay, boiling tube D will be put wet cotton wool. Yeah? So this is wet cotton wool, and then we put a seed on the surface of this wet cotton wool. Okay, so for A, boiling tube A, boiling tube B, and boiling tube C, these three boiling tube will be put in room temperature, while boiling tube D will be put into refrigerator that have temperature about 10 degrees Celsius. So that is the characteristic that exactly have a different condition of boiling tube to show whether the seed that put in each boiling tube can germinate or not. So after we have prepared all these 4 boiling tube, so we have to put aside for about 5 days. Yeah, so we have to put aside about 5 days, and then after 5 days, so we have to observe and record whether the seed germinate or not for every boiling tube. So the observation that we have made, we have to put or record it into the table, result table. Okay, the result table, okay, can be shown here. Okay, we can put or record the observation after 5 days into this table, result table. So we have test tube A, B, C, and D. Yeah, and then from here we have to put an information or the condition for every test tube, boiling tube, and then after 5 days, we based on the observation, we put the observation here. So based on the for each condition of test tube, okay, in test tube A, we have water, air, and suitable temperature. If we look back into boiling tube A, so we have wet cotton wool, means that we have water here, and then we have air, because this boiling tube is open, so air can enter into the boiling tube, and then suitable temperature, that is room temperature. So in boiling tube B, we have air, but we don't have water because we are using dry cotton wool, and we put into the room temperature. So we put here, we don't have water, we have air and suitable temperature. Okay, how about boiling tube C? Okay, boiling tube C using boil water. Why we are using boil water? And then on the surface of boil water, we put cooking oil. Okay, exactly why we are using boil water? Because boil water do not have any air inside. Means that no dissolved air inside the boil water. Yeah? And then why we put cooking oil? Okay, cooking oil is to make sure or to avoid or to prevent air from the atmosphere to enter and dissolve into the boiled water. So this method is exactly we want to eliminate air from the water and from the seed itself. So we want to know if seed can germinate without air. So this is the way on how we can terminate or expel the air in the water. And D, boiling tube D, we have water because wet cotton wool. We have air that can enter into the boiling tube, but we put into the cold temperature that is 10 degrees Celsius. So the info given here, okay, in C we have water, but we don't have air, suitable temperature. In D, we have water, we have air, but not suitable temperature. So based on the observation after 5 days, we can conclude, we can observe that, okay, okay, seed here, seed will germinate, okay, seed germinated, while in B, okay, does not germinate, does not germinated, and same with C and D, not germinated, and this one also not germinated. Yeah. So now from this one we know that in order for the seed to germinate, so seed required water, air, and suitable temperature. Without water, seed cannot germinate. Without air, seed cannot germinate. And without suitable temperature, seed cannot germinate. So this experiment shows that seed required all the condition, that is water, air, and suitable temperature. Okay, so that is the concept of seed germination, the condition that required by seed to germinate. So that's all. I hope that you can understand the concept of seed germination and what is the condition required for seed to germinate. So after this, we will discuss about the exercise given in the science activity book. So I hope that you will do the exercise given first, and then we will discuss the question next week. Okay, that's all for today. Thank you.